Hi everyone, welcome back. So this is part 2 from the chapter 2 of movement of substances. And for today, we are going to talk about osmosis. Okay, so in my previous video, I have already mentioned that I have split this chapter into 4 parts. And for this video, we are just going to talk about osmosis and the effects on plant cells and animal cells. Okay, so let's begin. This is the syllabus. All right. You must learn how to define osmosis. Okay. And you must investigate and describe the effects of osmosis on plant and animal tissues and cells. Okay. So because it's biology, you have to apply the concept of osmosis from a biological point of view. So let's begin. Okay. So this is the definition of osmosis. It is somewhat the same as diffusion. We're using the word net, okay, but it is no longer particles or molecules. It has to be water molecules, okay. Osmosis is purely talking about water molecules, and they also move from high to low, but we do not use the word concentration. We use the word water potential. Okay, because osmosis is a very special kind of movement, and so we are not allowed to use concentration. Okay, even though the concept is the same, but the way of writing, right, has to be different. So it is the same from high water potential to low water potential region. Okay, it is still the same down a water potential gradient. Okay, no longer concentration. All right, and osmosis has one extra stuff to talk about, which is across a partially permeable membrane. So this is the definition of osmosis. And everything that I highlighted is important, okay? So water potential, right, generally means the tendency of water molecules to move from one region to another. All right, you don't have to study this, but if you are confused, this is what water potential means. Okay, now osmosis, just like diffusion, will continue to occur until both regions are of equal water potential, and we use the word equilibrium to describe the two regions. Okay, they are the same. Osmosis does not require energy, just like diffusion. Okay, so just like diffusion, it is considered a passive process. And finally, which is different from diffusion, osmosis requires the presence of a membrane for it to occur. If there is no membrane, then there is no osmosis. Okay, so it's pretty unique in this way. And that's the reason why it applies a lot to cells, because cells always have membrane. Okay, so this confusion between water potential and concentration, right, is something that most students will encounter along the way, but actually it is pretty simple. They are opposite. Okay, so when we talk about a dilute solution, right, when we talk about the solution being very dilute, it actually means that the solution has low solute concentration. Okay, and because it has low solute concentration, the water potential is high. It is opposite. All right. On the other hand, if we use a term to describe a solution as concentrated, that means, right, it has high solute concentration and low water potential. Okay, so water potential and concentration, right, is actually opposite of each other. Okay, this table over here will tell you the difference, but it is generally what I'm trying to describe to you. Okay, it's a bit confusing, and so when you write, right, please be careful on how you use the word concentration and water potential. Okay, they are opposite. So let's use an example, right, of a situation. I have over here, right, I have region A and I have region B. Okay, they are separated by a partially permeable membrane. Okay, so over here, right, in region A, you can see, right, there are the ones in orange. It is sucrose molecule, which is a type of carbohydrate, and the rest in blue, right, is water. Okay, now in region B, right, you will see sucrose concentration also, but they are lesser. If you count, right, in region A, there is 1, 2, 3, 4, 5, 6, 7, 8, right? There are 8 sucrose molecule. And in region B, right, if you count, there are only 4. So before you actually answer the question, okay, you have to observe the diagram, and there are actually three questions that you need to take note of, okay? So I want you to note the difference in concentration between A and B, right, which I already described to you. Okay, so which one has higher concentration than which one? Okay, ready? Yeah, so A has higher concentration than B because A has more sucrose molecules as compared to B. A has 8 in this example and B has only 4. So in terms of concentration, right, A is more concentrated than B. Okay, now what if I ask you about water potential? Now, if you remember, right, concentration and water potential are opposite of each other, correct? So if A has higher concentration than B, it means that A has lower water potential than B. Okay, it is the opposite. All right. And because of the difference in water potential, right, which means that B is higher. Okay, I try to write the word H here, and A is lower in terms of water potential. What do you think? Okay, what do you think the water molecules will move? Now in this example, right, the sucrose cannot move because the sucrose cannot pass through the partially permeable membrane. However, water, being a very, very small molecule, they can pass. So the sucrose cannot pass, but the water can pass. And so if B has a higher water potential than A, it means that the water will move from B to A. Okay, like this. And when they move from B to A, after a while, you can see that the water level in A has risen and the water level in B drops. Okay, it is because of water molecules moving. Now, if you count the sucrose molecules in this slide, right, it is still the same. A still has 8, B still has 4, okay, because they cannot pass, but water can. And so you will create, right, a situation whereby the end result is that the water level is no longer the same. Okay, so this is what we call osmosis. All right, it is purely about the movement of water molecules. So how do we apply osmosis in living organisms? All right. Now this is an example of a plant cell that is soaked in a solution that is very concentrated, and so you can see, right, that the plant cell has shrunk, but the cell wall of the plant cell is still intact. Okay. We will talk about the scenarios in a while, but this is an example of osmosis happening in the plant cell, which causes the plant cell to lose water. So let's start with animal cells first. Okay. In a solution of high water potential, right, than the cell, which means the outside of the cell is high water potential than inside the cell, it means that water will enter the cell, okay, because water will move from high to low, right? So if water enters the cell, right, the cell will expand and the cell will finally burst, okay, because animal cells, right, do not have a cell wall. So if you put an animal cell in water or in a solution that is low, that is of high water potential, the cell will ultimately burst, okay, because water molecules will enter the cell non-stop. If you put an animal cell, right, in a solution that is the same water potential, in simple words, nothing will happen, okay, because there is no net movement. All right, there is no water moving in and out in a collective way. All right, so we say that there is no osmosis occurring. Okay, they are in equilibrium. And lastly, if we put an animal cell in very concentrated solution or in a solution of very low water potential, it means that the water potential in the cell, right, will is higher than the water potential outside the cell. And so water, right, will move out of the cell non-stop. They will continue to move out of the cell until the cell shrinks. And for red blood cells, right, they will end up being crenated. Okay, crenated means at the end of the day, right, the cell will look like will the cell will take the form of a spiky surface. So when you look at the cell that is with spikes coming out, right, yeah, it shows that the cell has lost water, and the cell is described as being crenated. But I think the word crenated is not so important, okay? But you need to understand that the cell will shrink. Now, on the other hand, plant cells will have the same movement, but the end result is not the same. Okay, if we put a plant cell in high water potential, all right, for example, water. Okay, water will enter the cell. Okay, the cell will continue to expand. Okay, and we see if you see over here, right, you see the word cell set. For plant cells, right, we need to describe the word cell set because the vacuole of a plant cell stores a lot of water. So the water, right, when we talk about comparing water potential for a plant cell, we have to compare between the environment and the vacuole. So you cannot just write plant cell, you have to write vacuole or cell set. Okay, this is very important because this is a keyword in biology. So water, right, will enter the vacuole. Okay, it will enter the vacuole, right, until the vacuole will expand and it will exert turgor pressure against the cell wall. So the vacuole will continue to expand and expand and expand, and the pressure inside the cell, right, will be very high. Okay, but the cell will not burst. Okay, so over here first, right, let me talk about it. The cell wall prevents the cell from bursting because the cell wall is of a certain rigidity. And so instead, compared to an animal cell which will burst, a plant cell won't burst and it will become very, very turgid. Okay, we use the word turgid to describe. And if the cell is very turgid, right, the whole plant, right, becomes very firm or hard. Okay, so when we describe a plant, we cannot use turgid or flaccid. We have to use hard or soft. All right. When we describe a cell, we describe a hard cell becomes turgid and a soft cell becomes flaccid, which you will see later. If we put a plant cell in a solution that is of equal water potential, again there is no net movement, so there is no osmosis, and so the two regions are in equilibrium. Now the opposite will occur if we place the plant cell in a solution of lower water potential. This time the water potential in the plant cell, right, is higher than the water potential outside. Okay, where in the plant cell? Again, we talk about cell set and vacuole. So water molecules will leave the vacuole. Okay, because the vacuole is losing water, the vacuole will start to shrink. And when the vacuole shrinks, right, it will draw the cell surface membrane, the cell membrane inwards. Okay, it will draw the cell membrane inwards, and then the cell will become soft. But we don't use the word soft, we use the word flaccid to describe the cell. Okay, you can also use the word plasmolyzed, but flaccid is more important. All right. Now again, you will not see the collapse of the whole cell because of the rigid cell wall. So in this diagram, right, you can see that because even though the cell is losing water, generally the cell still retains the rectangular shape. Okay, and because the cell becomes flaccid, right, the whole plant tissue becomes soft. Okay, so this is what happens when we apply cells to solutions that is of higher or lower water potential. Okay, very important. So what is turgor pressure in plant cells? Okay, turgor pressure, right, is pressure exerted by water against the cell wall of a plant cell. So turgor pressure, right, will affect the turgidity. High turgor pressure, high turgidity. Low turgor pressure, low turgidity. Okay. Now this is very important in plant cells. Okay, we don't use turgor pressure to describe animal cells. Okay, because yeah, they will burst. But because they have a cell wall, right? This turgor pressure is important in plants. Okay, one example is that the young shoots or seedlings, right? Because plants got no skeleton to remain upright. So by having a high turgor pressure, right, they can remain firm and upright when they are growing. Okay, so they can grow in a upward movement. Okay, gut cells, right, which we'll study later, gut cells can manipulate turgor pressure, and this manipulation, right, actually opens or closes the stoma or the stomata. Okay, if the turgor pressure of a plant cell, right, is lower than usual, the plant will wilt. Okay, the plant will droop, which is not good for the plant. So that's it. All right, this is how osmosis occurs in animal and plant cells. So if you have any questions, please ask. Okay, but yeah, this is the end of part 2. Thank you. Bye bye.